you so much for being here for today's seminar. This is a hybrid event, so we have folks in the room and folks online. So thank you everybody for being here. For those folks that are online, just a reminder, we do have your cameras and screen shares and mics turned off, but we do hope that you ask any questions into the chat and we'll get to them at the end. For folks in the room, we will also get to your questions at the end, but we will need you to have a mic. So you have a couple of options. You can raise your hand if you have a question, or you can go to the mic stand up there so I don't have to run all the way to the top. If you have a question, you can ask it there. But I just wanted to say thank you for being here. My name is Cinnamon Moffett. I'm the Research Program Manager here at Oregon State University's Hatfield Marine Science Center, located in Newport, Oregon. I will be your host. We also have Sana Banks, who is going to be the host for the online folks. So folks online, if you have any questions, you can go ahead. Technical issues, let Sana know so she can let me know in the room and we can adjust as needed. I wanted to make a couple of quick announcements. Tonight, we actually have a Science on Tap. So if you are interested and you haven't got enough after Sam's talk today, please come on back at 6 when Scott Baker is going to be talking about historic whaling. And that's my phone, and I did one of those horrible things. All right, go ahead and turn your phones off, everyone. That was my fault. So Scott Baker is going to be talking to us at 6, and there will be a social before where the cafe will be serving beer and snacks, and that will start at 5 today. So if you're interested, come on back. Next week, we will have another seminar, same place, same time, but this will be a fully remote seminar next week. We will not have any on-site option. Our speaker is going to be coming to us from California and is going to be talking about trying to decide, should I stay or should I go? And looking at some genetics along with that, looking at monarch butterflies, steelhead, and salmon. So excited to have Will come and talk to us about that. For everybody, this event is being recorded. So in a few days, it will be on the past seminar page. So you're feel free to share it with others, or if you need to watch it again because we missed something, feel free to do that. And if you need more information about any of our events, you can go to the Hatfield homepage, scroll to the bottom. There's a calendar of events there with all the login information that you might need. But while you're all here today, I wanted to introduce you to Sam, is going to be our speaker. Sam completed his PhD at the University of California and studied sea anemones and their mutualistic relationship with algae. He is a postdoctoral scholar at Oregon State University, where he continues to study sea anemones as a model organism to better understand coral symbiosis. And I just found out that Sam will be here as an instructor this summer. So we're excited to have Sam here. Sam, go ahead and take it away. Turn on your mic.
All right, can everyone hear me? Okay. I'm so excited to be here today. This is amazing and beautiful place. This is my first time here, so this is very exciting. I just moved to Oregon in January. Yeah, I feel very welcome. Thank you for that wonderful introduction. So I'm going to get right into it. This is a little bit of the work I did during my dissertation at UC Irvine. And I studied sea anemones, as you might see. I have it on my shirt. I have it on the screen. And today I'm going to be focusing on the trophic and non-trophic interactions of the sea anemones that we see in the rocky intertidal zone. So I started out this journey thinking about exclusively trophic interactions. What are sea anemones eating? What is eating sea anemones, right? But it led me, and I'll show it through the story that I have here, to thinking about how non-consumptive interactions between sea anemones and other invertebrates in the rocky intertidal zone affect each other. Okay, so when we think about the rocky intertidal food web, this is from a paper by Bruce Mingy. We think about consumers and those who are consumed, the predators and the prey. So here we have large carnivores at the top. Let me see if my mouse, there we go. Large carnivores at the top. We have mobile herbivores. We have small carnivores. This is, of course, a very simplistic diagram here. But in my mind, when I see this, I see a problem. There are no sea anemones. We don't know how sea anemones fit into this food web very well. And it's because sea anemones can be complex in their diets because they're eating things, right? They're carnivores, but they also kind of act like plants. They have algal symbionts that live inside their tissue that photosynthesize. So they have multiple ways to get energy. And so a lot of times they're overlooked. And even though they're overlooked, they're very common in the rocky intertidal zone. As you might know, if you've been out to the tide pools, you look in tide pools and you're going to sometimes see hundreds of sea anemones in these tide pools. These are all Anthopleura xanthogrammica with a few Anthopleura elongantissima that I'll be talking about today in here. But the traditional way we think about these sea anemones is that they're just sitting there like plants, they're photosynthesizing. They're not these ravenous carnivores that they actually are. They eat what we traditionally think that they eat are mussels, and you can see those mussels kind of surrounding this tide pool. But what I'm going to show today is that their diet is highly diverse. It doesn't just include these mussels. The mussels are actually a very, very small part of their diet. So I mentioned this relationship between algae and sea anemone, right? And how these sea anemones can benefit from photosynthesis of these algae that give them carbon from photosynthesis. So here I'm showing the two nutritional pathways that they have. They have a heterotrophic pathway that has to do with eating things because they are predatory animals that have tentacles and nematocysts that grab onto prey and pull it into their mouth. They also have these algal symbionts, or at least most of the species that we see in the intertidal zone do. So they're exchanging nitrogen with these algal symbionts for carbon in the form of sugars and lipids. So the species that we see along the west coast of North America are these 5 in the genus Anthopleura. So these are all the ones in the genus Anthopleura that can be found in the rocky intertidal zone. We have Anthopleura artemisia, which we do see in Oregon. Then we have Anthopleura murrayi, which actually might be a different species. We're still looking into that, but that's only in Southern California and Baja. And then we have 3 symbiotic species that I am focusing on, or at least I focused on for my dissertation. We have Anthopleura elongantissima, Anthopleura sola, and Anthopleura xanthogrammica. You might not see Anthopleura sola up here, but we do see Anthopleura elongantissima and Anthopleura xanthogrammica. So these 3 are the ones I focused on. They're the symbiotic ones. There's a lot of size variation among the 3 species. So Anthopleura elongantissima, it reproduces asexually by splitting and creates new individuals and creates these large clonal mats. So that's a big part of their ecology, is that they stay pretty small. Anthopleura sola is a larger species. It only reproduces sexually. You can find it individually. And then Anthopleura xanthogrammica is similar to Anthopleura sola, but it's even larger. And my question when I started my dissertation, which morphed into something different, but when I started, I thought about these 3 different species that are along the California coast. And the California coast is really unique because we have all 3 of these species overlapping in their range in Central California. We have Anthopleura elongantissima along the entire coast, that small species. And then we have Anthopleura sola coming up from the southern region, and we see that going up to the northern end of California. And then we have Anthopleura xanthogrammica in the green here, coming down from Oregon all the way up to Alaska, down to Central California. We can even find some xanthogrammica down in Mexico, but it's pretty rare. They really follow upwelling zones as what xanthogrammica distribution follows. So I was really interested, why do we have all 3 of these species existing in this central region? Are they having, as their niche partitioning going on? And my first thought was, okay, their diet could play a role because they're very different sizes. Maybe they're eating different things. So these are our Oregonians up here. And this is the species that's in California, but it is moving up north. So it's slowly making its way north as the climate warms. So how do these sea anemones eat, right? They have these tentacles that just lie in wait. They're passive suspension feeders that wait for things to come in contact with those tentacles. Then the nematocysts shoot out and sting whatever they come in contact with. The tentacles wrap around that prey item and push it to the mouth. and then it's moved into the gastrovascular cavity of these sea anemones. And the diet of these sea anemones, we know a little bit because some people in the 1980s, mostly Ken Sebens, here in Oregon and Washington, looked at the diets of these sea anemones. He went out to the intertidal zone and collected hundreds and hundreds of these sea anemones and then looked inside their gastrovascular cavities for what they were eating. This method was pretty rough because we only get about 5% of those sea anemones that have anything in their gastrovascular cavity. So it's very poor sampling. You can sample hundreds and hundreds of anemones and only get a few with prey items. So I took a different approach to this. And I also was interested in, okay, we can find these things in the gastrovascular cavity, but are they digested? Are they kept? Is something else going on? So this is just a pretty picture showing you the tentacles of the sea anemone and the mouth. So this is the mouth opening up. This is Anthopleura sola, the sunburst anemone, as you can probably see where it gets its name from. But it will grab onto things and put them in its mouth. All right, so the first question I had posed at this time during my dissertation was just how do sea anemones in the genus Anthopleura fit into the rocky intertidal food web? And I thought, okay, I'm just going to go after trophic interactions here. So my null hypothesis for this part of the talk here is that sea anemones will consume anything that they can capture, any animal that they can capture, showing no preference among them, right? And the reason why I thought this is from observations from the field and observations from others. So if you go out and you look, which I'm sure many of you have, at sea anemones in the rocky intertidal zone, you can see things on their oral disc that they've eaten and are then egesting, right? So this is a sea urchin test. This sea urchin was unlucky and fell into the sea anemone and was digested. And that's the test being spit out after it's been digested. So we find lots of things like that. We can find mussel shells, all kinds of different things that these sea anemones are egesting after they've been digested. So that's one way we can figure out what's going on. But then they also have some really odd things that I've seen them eating. And this is what got me interested in these trophic interactions. So this is a rabbit that ended up in the rocky intertidal zone. We aren't really sure how it ended up there, but it's being eaten by two sea anemones. So here's the head up here. You can't really see the head, but it's being eaten by a sea anemone up here. And then the foot down here is being eaten by another sea anemone. They're kind of playing tug of war on this rabbit, eating it. And so what's really unique about sea anemones is that they don't have to have the whole animal inside of them to digest what they have. They just have to have a part of it, they digest it, and then spit out whatever's left, right? Other observers have noticed some interesting things like this. This is from a paper Guy et al. 2014, where they found cormorant chicks that would fall off of cliffs that would end up in sea anemones. And so the sea anemone would just digest the head and then get rid of the rest, right? So they have all kinds of the crazy trophic interactions. But a lot of these are scavenging events, right? They're just eating the things that are dead that end up in the rocky intertidal zone. What I was really interested in were living invertebrates that they're interacting with on a regular basis. Are they consuming those, or are they just serving as scavengers in these systems? So the way I got at this, I couldn't necessarily go out in the field and watch what they're eating all the time because the waves are crashing. I've tried it a couple times. I've tried to set up cameras, but it is very hard. So I brought them into the lab to do these feeding trials so I could control different aspects. So this is a quick video here. What we did is we fed these sea anemones invertebrates, right, that were common in the rocky intertidal zone. And then we would watch how they would respond. So in this case, I'm showing you two that I've given this anemone. During my trials, I only gave them one at a time to make the analysis simpler, so it's more straightforward. But you can see here on the left-hand side, we have a mussel that I put in, and then on the right-hand side, we have a snail, a Tegula phoenobralis. And so you can see the anemone kept that mussel and got rid of the snail. So this is the setup we had. We had these anemones in the lab, let them settle in these containers that we had. Then we would put them under GoPro cameras and we would watch their behavior with different prey items that we would give them. We gave them both living and dead prey items, getting at that scavenger question, and whether they had any preference or if they just consumed everything that we gave them, right? And we followed their behavior for 2.5 hours after we fed them these items. So we were able to record behavior of these sea anemones and whether or not these items were just taken in and then spit out, or if they were always kept and digested. So these are the results from that. And I'm showing you, I used all 3 species of sea anemone that I've been talking about. And I was expecting to find different preferences for different sea anemone species, but turns out they all respond the same way. So we didn't see any difference in behavior. So the data I'm showing you here is all 3 of the species combined. On the x-axis, we have the different species of invertebrates that we offered them. And then on the y-axis, we have the proportion. So the proportion is either the proportion retained in the dark bars or the proportion digested in the empty bars. First of all, what we noticed is that 98% of all the dead items were retained and digested. So they like everything. Anything that's dead, they'll eat it and digest it. And that is that kind of supports the idea that they're definitely scavengers in that regard. But the living items are all the data that we have here. So these are all the things that were alive that we offered these sea anemones. And what we found, and the reason why we have these dark and light bars, is that some things were retained, but not all things were digested. So a few examples of this. Snails, these 3 snail species that we included, they're sometimes retained. So you can see that these 2 species, Apunctulata and Circumtexta, they were sometimes retained. It wasn't significantly different from no preference, but they were sometimes retained, but they were never digested. We would find them egested by the anemone, and they were perfectly fine and alive. They'd just be crawling around afterwards. So they can survive inside the gastrovascular cavity of these sea anemones. The one interesting point is this Tegula fenebralis was almost never retained and it was never digested. And I'll come back to that on the next slide, but that's the oddball right here. This species is strange. Okay, so what about mussels? The thing that we classically think of these sea anemones eating, right? So mussels they're not moving much, right? They're usually attached to the substrate. The way they end up inside of sea anemones is they get broken off, right? Wave exposure, things like that, disturbance. They get broken off and they end up in the sea anemones. We generally think that if a sea anemone gets a hold of a mussel, it's doomed, right? There's no chance. Well, it turns out sea anemones sometimes don't keep mussels, even though they have no way of getting away. Sometimes sea anemones will just reject them straight out. So they're not always accepted, which is interesting. And then some of those that are kept within the gastrovascular cavity, the mussel can keep closed and escape by just being egested, and they're perfectly alive just because they can stay sealed up. It's pretty interesting. The snails are probably doing a similar thing. And then lastly, we have our 2 crustacean species. We have the hermit crab here and a shore crab. And they can escape by crawling away. One of the coolest things is that hermit crabs can leave their shell behind. So if their shell gets grabbed by an anemone, they'll bail and be like, bye. They'll leave their shell and run away. And sometimes they can escape that way. But if the anemone can grab them and hang on, they don't stand a chance. They always get digested. And yeah, they're in danger if they're near sea anemones. So that Tegula fenebralis, this species here, this herbivorous snail, we thought it was really unique that the sea anemones almost never keep those snails. They almost always get rid of them right away. And so we went into that in more detail. What we did is we manipulated these snails. We had different conditions here on the y-axis. We have unmanipulated at the top, which is the same data as you saw previously. And then as you go down, we have happy living snails, and then our gradient goes to dead snails here. So I'm including all 3 of the sea anemone species here, which can make this graph a little daunting, but it's showing you that there is a difference in behavior of these sea anemones for Tegula, which is pretty interesting in some of these treatments. So if we go from the top, which is the unmanipulated living snails, they almost always reject. So this is the proportion retained. So if we're on the right-hand side, they're accepting the items. If we're on the left-hand side, they're rejecting the items. So almost all of them, almost all of our sea anemones rejected tegula that were unmanipulated. Then we said, okay, maybe it's the operculum, the little door that closes the snail, right? Maybe that's it. So we removed that to see what would happen. And they still almost always rejected them, right? Then we're like, okay, what about if we just gave them a shell, right, of a tegula? Maybe it's the shell. Maybe they don't like the shell. So we filled a shell with silicone and then covered it back up with the operculum. And then we fed it to the sea anemones. And what we found is that apparently Anthopleura xanthogrammica can tell that it does not want this, right? It says, no, this is fake. But Anthopleura sola and Anthopleura elongatissima, we see no preference in either direction. They could take it or leave it. So there's something to do with that living snail in there that is having an effect. Then if we look at more manipulations as we go down, we have a cracked shell. So mostly they're kind of take it or leave it here. And then as we go down, no shell and then dead. They will always eat them if they're dead, and they're happy to. So it's not about the snail's tissue or anything like that. There's something else going on. So this made us think that there's some kind of chemical defense going on. So the next thing we looked at was the behavior of these sea anemones. Since we recorded video, we could look at how long these snails were in the gastrovascular cavity of the sea anemones. And what we found is that any living snail, so these three here, no operculum, cracked shell, and unmanipulated, they were all kept for only an average of about 17 minutes by these sea anemones. And then our mimic, which had no living snail in it, was kept for much longer periods, about 85 minutes on average. So there's something to do with that living snail inside that's making them not want it, right? So we're guessing that this is evidence for a chemical defense. And if anyone's interested in trying to figure out what this chemical defense is, I'd be happy to work with you. We still don't know what this is. We're just getting into this and thinking about what this could be. But it's especially telling, in my opinion, that the no operculum was egested faster than the unmanipulated treatment. So maybe that chemical defense is released faster when you don't have a closed door in front of the snail. Okay, so going back to our null hypothesis saying that sea anemones will consume all animals regardless of what they are. That's not true, right? What we found is that snails can always survive, even if they don't have this chemical defense, like our two carnivorous snails. We didn't find evidence for a chemical defense in them. They just stay closed and get spit out, and they're fine. They can survive. The crustaceans, they can't escape the tentacles, but they're always digested if they can't escape them. And then it's interesting that mussels are not always eaten because that's the classical thing that we think that they eat. And then finally, Tegula fenebralis may be chemically defended against predation by these sea anemones. So this got me thinking, I was really interested in trophic interactions. What if there is an interaction here that's non-trophic? And what I kept seeing over and over again when I was out in the field is sea anemones that had snails around them. And so I would go out and I would see something like this. I would see, this is Anthopleura sola here in a tide pool. And then I would see Tegula fenebralis and other snails just surrounding that anemone. You can see here's an even more extreme example. This is Anthopleura elongatissima, the aggregating anemone. It's covered in debris here. But you can see little limpets that all congregate around the edges of these sea anemones. And so I started thinking, what's going on here? Why do we have this association? So my next question was, could sea anemones provide habitat for mobile invertebrates? So there are a lot of other systems where we see organisms providing microhabitat and increasing biodiversity, right, through habitat modification. We have a lot of examples, terrestrial examples that I'm not super versed in, but we have Douglas firs that provide all kinds of microhabitats for organisms. We have bromeliads that provide these really unique microhabitats for all kinds of aquatic animals. But we also have symbioses or mutualisms that form these associations that allow for biodiversity to build around them. For example, we have estuaries where we have grass that associates with mussels in this mutualism and then attracts all kinds of other invertebrates and increases biodiversity in a mostly soft sediment habitat where having hard sediment created. We also have the classic example of coral reefs with their algae and the coral working together that create these unique habitats and trophic interactions and create huge biodiversity. I thought maybe this is happening on a much smaller scale in the system, looking at these sea anemones and the mutualism with their algal symbionts. And I'll talk about why I think this is all interconnected. So if we look at sea anemones out in the intertidal zone, over here, we have a crack in the intertidal zone. This is a kind of angled area in the upper intertidal zone at Corona del Mar. And I would continually see these areas that were super dry, nothing going on, very few invertebrates there. And then I would see these colonies of aggregating anemones that had invertebrates that would congregate all along them. And if you look along the edges, it's darker here. That's all water being released by those sea anemones during low tide. And we know this happens. Brian Bingham in 2011 showed that the sea anemones release water at low tide to avoid overheating and desiccation stress. And so they're kind of like balloons. During high tide, they blow up, right? And during low tide, they slowly release the water. And so if you go out, you'll see there's usually pools of water, even if they're not in a tide pool, around sea anemones. And so these sea anemones, unlike their non-symbiotic brethren who are living in substrate or under rocks or areas where the desiccation is not a problem, symbiotic species with these algae live in really open habitats where they can photosynthesize and take advantage of that nutritional pathway, right? And so we usually see them in these habitats that are really open. So they've come up with this adaptation to survive here by releasing water. And so I thought maybe this mutualism is cascading into effects on facilitation and increasing diversity of these mobile invertebrates. And if you look really close, again, you can see all those invertebrates kind of congregating around there. And so no one had ever looked at this interaction before, and so we decided to investigate it. So the idea here, the hypothesis, is that this mutualism has resulted in this adaptation to keep the mutualism that is temperature sensitive in check. They'll release the water, it'll reduce the temperature and keep them from desiccating. And therefore, other invertebrates will take advantage of this habitat. We did some experimental manipulations at 3 sites in California, from south to north here: Corona del Mar, Colo Point, and Rancho Marino. All 3 of these sites have a lot of sea anemones in the upper Rocky Intertidal Zone, and they have very different assemblages of invertebrates. That's why we chose these 3. So our 3 hypotheses. The first one is that temperature and desiccation will be lower in sea anemone habitat, trying to continue to confirm what Bingham found. Hypothesis 2, that mobile invertebrate richness and biomass will be higher in that sea anemone habitat than in adjacent habitat that's matched. And then hypothesis 3 is most important, experimenting, manipulating that habitat to see if it's just the underlying habitat that's having an effect, or if it's actually the sea anemones that are impacting the community. So this is how we looked at temperature really simply. We put out data loggers, both in sea anemone habitat here on the right, and then we had it on bare rock. So this is one of our examples. And this is what we found. We confirmed Bingham's result. This is the maximum daily temperature on the y-axis. And then on the x-axis, we have our different treatments, or I'm sorry, not our different treatments, but our different areas, habitats we are measuring. And then throughout the rest of the talk, the gray is going to be the adjacent rock. And then the bluish color is going to be the sea anemone habitat. And so we found that the maximum daily temperature during our lowest extreme tides was higher in the adjacent rock habitat than it was in the sea anemone habitat. And I just want to point out that these two habitat types were only 10 to 50 centimeters apart at most. They were matched in their type, their angle, and everything. So we try to keep it as consistent as possible for both this and the experimental section next. So the temperature is higher on the adjacent rock habitat. If we look at relative desiccation stress, we can see that the water loss, so the way we did this is we put out little cardboard discs that were soaked in water, and we measured the difference in the evaporated water between the beginning of our time out in the field and the end after about a 4-hour low tide period. And what we found is that the water loss was higher on the adjacent rock habitat than it was in the sea anemone habitat. So temperature is lower in that sea anemone habitat, and potential relative desiccation would be lower in the sea anemone habitat because they're constantly releasing that water. Okay, so who's in these habitats, right? So part of this survey and these observational experiment or observational work that I was doing was looking at, oh no. Okay, well, the species are gone. There's a conversion issue. Okay, I'll just have to describe them to you. So we have here on the y-axis just a bunch of species that aren't there anymore, and then on the x-axis we have the proportion in each habitat. And so as you go towards 1, more positive, that means it's in sea anemone habitat. And then as you go to the negative numbers here, -1 would be always prefers the adjacent rock habitat. And so out of the 11 species that we looked at, we found that 2 of them were a preferred adjacent rock habitat. And then we found 7 of them preferred sea anemone habitat. 2 of them had no preference. These 2 up here, I won't talk about the specific species, but these are 2 limpets. And please come to me afterwards if you're interested in the specific species. I can show you the figure. These 2 species here are specialists in the upper intertidal zone. They're limpets that kind of have grazing scars that they sit down into and survive really well in the upper intertidal zone. These species down here are usually species that we don't see in the upper rocky intertidal zone. But when we have these sea anemones up there, we can find them under the sea anemones. Some interesting ones out of these that prefer sea anemone habitat. There's one that's a parasite, Epiptonium tinctum, the wentletrap snail. It eats sea anemones, and so it'll hide underneath the sea anemone during low tide to survive, and then during high tide it comes out and eats the sea anemone. It's a pretty cool interaction. And then we also have other species that don't have any trophic interactions with these sea anemones. A lot of them are snails, and one of them here at the bottom, which is punctulata, Acanthonucella punctulata. It is a predatory snail that is found exclusively under sea anemones in this upper intertidal habitat. It'll hide there during low tides, and then it comes out and looks for prey during high tides. So the next thing I did is I went out and I experimentally manipulated these sea anemones. So here I am on an early morning tide, removing a sea anemone and moving it to a new location. And so moving sea anemones is not as easy as you might think, or maybe you know. Sea anemones don't like to behave. But we try to do our best to make them behave. We did different experimental treatments. We did an anemone control, we left that alone, and then we did a rock control, and that's again directly adjacent and matched in habitat. Then we did a removal and addition, and these were paired. So we went in, we very carefully removed the sea anemone from the substrate, and then we moved it to the addition area. To keep it there, we used mesh, and we put that mesh on top of the sea anemone and to hold it against the substrate, and we left it for 48 hours to attach. After 48 hours, we would come back and we would take the mesh away and just hope that it would stay. And sometimes it did. So we got some results here. But you'll see the sample sizes for the addition treatment are much smaller than the removal. So what did we find when we manipulated these sea anemones? On the y-axis here, we have invertebrate richness in a very small area underneath these sea anemones. So you can see the numbers are pretty tiny. We found that the rock control was lower than if we look all the way to the right here, the anemone control. So that was consistent during our observational period and during our experimental period. But what we were really interested in is when we take the rock control and we add a sea anemone, what happens? And what we found is only after 4 weeks of this sea anemone being there, they already had a higher invertebrate richness. If we look at the anemone removal and anemone control now, so if we take an anemone and then we remove it, what does it look like? Our richness decreases when we remove the anemone. So we found evidence for richness increasing when sea anemones were present and decreasing when they were removed. But what about biomass? So biomass is even more evident here. This is the same exact setup. You can see rock control is always much lower than anemone control. And if we look and see what if we add a sea anemone to the rock control, we see an increase in biomass after those 4 weeks. And then the same thing, if we go in and we remove an anemone, we see a decrease. So there's evidence that these sea anemones are increasing richness in biomass where they're present. So again, just summarizing these results, we have this mutualism between the algae and the sea anemone. It is a delicate relationship that is dependent on temperature, right? And so the sea anemone has to keep itself cooler in those really hot, dry environments. So it has this adaptation to release water during low tide. It reduces stressors, and then those mobile invertebrates take advantage of that behavior and congregate around those sea anemones. So these sea anemones can play a facilitative role in the upper intertidal zone. So going back to my original thoughts of thinking about these trophic interactions and then how it flowed into thinking about these non-trophic interactions. I was really interested in this mutualism, but then how does this mutualism affect the community? Well, number one, sea anemones are carnivores. They're eating a lot of things, but they aren't eating everything. And that was really interesting to find. They are picky eaters. They're not always eating everything that they come in contact with. They also have non-trophic interactions with other mobile invertebrates that we originally thought were eaten by these sea anemones, but actually taking advantage of these sea anemones as microhabitat. And so they fill a facilitative role in that case. So they can change the rocky intertidal zone by both non-trophic and trophic interactions. So if we revisit this paper by Mingi, thinking about the food web, okay, I added a sea anemone up there in the upper right-hand corner. How are they affecting this community? Well, we don't have everything here, but number one, they are eating filter feeders, at least mussels that they come in contact with. They are eating some of them. They are eating crustaceans, or necessarily included on here. So there is a consumptive effect, but they're also having a facilitative effect on these other invertebrates. All the snails, all the limpets, all the chitons are benefiting from these sea anemones, at least in the upper rocky intertidal zone in these areas. So we have a new understanding of how these fit into the food web. And with that, I'd like to thank all my funding sources throughout my time at UCI and my dissertation. And I'd like to acknowledge everyone who helped me with this research, including all the undergrads that I worked with, graduate students, and everyone else, all of my friends. And now I just wanted to talk about a little bit of the research that I'm doing now, because I'm kind of switching gears a little bit. Less ecology now, and now I'm working on physiology. I'm working in Virginia Weiss's lab at the main campus at Oregon State. These are Aptasia. So they are a model system for coral. They host algal symbionts just like coral do. And you can see on the left-hand side here and the right-hand side, they look different, right? The ones on the left-hand side have no algal symbionts, so they're kind of like bleached coral. And the ones on the right-hand side have those algal symbionts. And this is one of the main reasons why this system is really unique and great for experimental manipulation, is that we can remove the symbiont and then see how these cnidarians respond, just like coral, except they're a lot easier to keep in the lab than coral. So I'm going to be working on a lot of different things in the lab, including development of these development of asexual clones of these sea anemones. We're going to be working on sexual reproduction in these sea anemones, trying to settle their larvae, because that's particularly hard in this species. And we're going to be looking at symbiont shuffling and physiological aspects of the relationship. And here they are eating brain shrimp, which I just think is beautiful. Okay, and with that, I'm a little early, but I'll take some questions. You're welcome.
All right, thank you so much, Sam. So are there any questions online? We'll start there. No questions online. How about questions in the room? Yeah, Sarah. For folks online, I gotta walk up some stairs. Hang in there with me. Oh, that mic needs to be turned on, Sarah. So if you flip that one on, that would be great. Thank you.
Thanks for coming to talk to us. I'm excited you're going to be an instructor out here this summer. I had a question about the tegula. So they maybe have some sort of chemical defense. And let's see, if you put a live one in that's fully intact, doesn't digest it, but that's because it's all closed up. And then if you put one in there that's been slightly manipulated, it's like it tastes, you think, it tastes its yucky chemicals and it spits it out, but it'll eat a dead one. So are those chemicals not active unless the tegula is alive? Do you know, are there examples of chemical defenses that are like inactive when they're just in the body and not turned on until this event?
That's a great question, and that's something I want to answer. I wish I was a chemist. I'm not, and all I can do is, as an ecologist, and what's amazing is this came out of a project from an undergrad who is doing this, right? And so we tried to get it at every angle that we could with the resources that we had. And now, I mean, I would love to continue this and bring it with me, but yeah, I really don't know. I don't know if it's a chemical they're producing. I don't know if it could be something else. Yeah, I'd love any input.
Good. Future student projects.
Exactly. Yeah. We're going to be working on it a little bit this summer at the Marine Lab with the course that we're teaching. So we're going to extend some of this work because it's very friendly for simple undergraduate projects that they can carry out in a short period of time and get really neat results.
Nice. Good.
And have fun with the anemones.
All right. Questions online yet?
Yes, John Chapman has a three-part question here. The first part, is any information on the aggregating gastropods growing faster than other gastropods?
The aggregating gastropods, the ones that hang out next to the sea anemones?
I believe so.
Okay.
Yes.
Yeah, I'm not sure. I know that some of the gastropods that we see around these sea anemones, again, we only see them in the lower intertidal. We only see them around the sea anemones in the upper intertidal most of the time. So it could be that they're kind of escaping some of the competition that they have in the lower intertidal with conspecifics, and they could therefore be extending up there and be doing better than other ones would be if they were just exposed during low tides.
Okay, he also just had a question about, he just said, what about commensal amphipods? I'm thinking what he means is, were they present with the aggregates? John, you can
Yes.
also be unmuted. Just let me know if you want to be.
Let me know if you want to what? Oh, got it. Okay. We did see quite a few amphipods and isopods. I didn't include those in that original species group, but the number of species that we found under these sea anemones was something like 60 or 70 at least that were regularly there. Amphipods were one of them. We saw them quite a bit. We never went into trying to identify different species of amphipods. We just included them as amphipods, and we found some isopods. There are also amphipods that can be symbiotic that live in the gastrovascular cavity of these sea anemones, which just blows my mind. But yes, there are some amphipods that live underneath the edges of these sea anemones. They're very common there.
And the third part is, I think he's referring to what you just mentioned, because he's asking, are they parasites? So he must be mentioning the ones that live with the anemones.
Yeah, it could be that they eat the mucus from the sea anemone as well, because they generate copious amounts of mucus and mucus rings. And that's usually where we'll find them. And so they might be eating some of that. They might be using it as protective microhabitat, or maybe they're partially parasitic and inside, at least the ones that are inside the sea anemone could possibly be eating the things that are inside that sea anemone in the gastrovascular cavity, which no one's followed up on that work. That's really interesting. Yeah.
Cool. I think we have a question in the room. I'm going to hand this to each other.
A great presentation. I actually have 3 questions. One is, can you name the summer course that you're teaching this summer?
Well, I can't remember the number now. It's killing me because I just worked it out with Nate this morning,
Yes.
over the last couple of days.
So as the academic program manager here, I'll tell you, it is marine and estuarine invertebrate zoology.
Yes.
In our session 4 for summer session. So let folks know online as well as in the room. My question related to your work. Really fascinating finding about the snails, about Tegula. I'm wondering if you could elaborate a little bit on sea anemone's digestive tract, and have you looked at any analogous situations similar with terrestrial snails as far as a defense mechanism? Might it be not only chemical but also mechanical, similar to, say, hagfish slime?
Yeah, we have thought about that. So sea anemones are built, we like to say, like a trash can. That's how I describe it. So everything goes in one opening, and everything comes out one opening. So it's both a mouth and an anus in that respect. But they just kind of have a bag. They do have partitioning in that bag where we have mesenteries, where different prey can be in different areas. But all digestion is happening in one area, and they digest things fairly quickly. And if things aren't digested, they just egest them half digested. So they bring things in, they digest them between about 3 hours to 2 days at most, depending on the size of the item. But they won't keep it in there any longer than that. With mechanical things, no, I have not looked in terrestrial snails to see defenses. However, with other sea anemones, I have looked into chemical defenses of other animals that end up in sea anemones. There is aplysia, can release an ink that is aversive to these sea anemones in particular, and they will spit out aplysia. They won't digest them in a similar way, but that's much more obvious. You can see that purple ink, and so it's clear. For the mechanical, thinking about the mucus, we don't see any mucus when these snails are released. We haven't seen any evidence for that, but it's possible. What we see, it's kind of incredible. They spit them out onto the mouth, and the snail will crawl across the anemone's oral disc and just fall onto the substrate and crawl away. So what could be happening, I mean, it could be that the snail doesn't crawl out of the gastrovascular cavity. We don't see any evidence for that. When it's egested, it's completely closed. But it could be that they open up in there. Maybe they don't like the feeling of their foot, the gastropod's foot moving in their gastrovascular cavity. Could be that, but we don't have, we need to be able to see in there to see if that's going on. Yeah, great questions. Yeah.
Any other questions online, Sana?
This is from Parker Lund. He'd like to know, do the Aptasia also engage in water expressing behavior when exposed to the air, given that they're algal symbionts?
Yeah, so Aptasia are very different. They are a subtidal species. They aren't in the intertidal zone. They live on mangrove roots most of the time, and they're in tropical areas. If they're exposed to the air for any period of time, they'll dry out. They're like little bags of mucus. They're not nearly as tough as Anthopleura is, and so yeah, they don't show that same behavior, and we don't see the species diversity around them or anything like that. We do see like a lot of parasites with Aptasia, like pycnogonids and sea spiders that will attach to them. There are a lot of sea spiders that attach to Anthopleura as well. So that's the same between those two species, but yeah, very different species.
Any other questions in the room?
There's one more question. So Aptasia, can you maybe elaborate a little bit more on some of the questions that you're looking at with this new project?
Yeah. So one thing that we're looking at right now is these adult Aptasia. The Aptasia, they're, about 1 inch to 2 inches tall, pretty tiny. And we have gotten into this idea of development in this species that's symbiotic. And so these sea anemones, they leave, so Anthopleura, elegantissima, splits in half. It does this fission thing where it rips itself in half and produces 2 sea anemones. Aptasia produces these little tiny bits of tissue that it leaves behind. And they're just lumps of tissue. And those lumps of tissue then slowly develop into full-size adult anemones over time, right? So recently our lab has really been focusing on this asexual reproduction and development in these anemones. And it has a lot of parallels to coral in that they're growing, it's new growth. And so one thing that we're interested in is how do those symbionts proliferate through new growing tissue? And so we've been using those pedal lacerates as a way to think about, okay, these lumps of tissue are organizing themselves with these algal symbionts. How do they organize the algal symbionts, and how do those algal symbionts proliferate throughout that tissue as it grows? And so with the Aptasia system, we can use both the adults, which are not having this rapid growth going on, and those pedal lacerates that are having this rapid growth, and compare proliferation with different species of algal symbiont is the big thing that we're focusing on right now, is different strains and genera. We have tons of genera of symbionts that can live in these Aptasia. So yeah.
All right, last call for questions online in the room. All right, I think it's time to thank Sam and let him off the hook. All right, for everybody in the room, I hope we see you either next week online or maybe even at Science on Tap tonight. Thanks for being here. And one more time, thank you so much, Sam. We appreciate it.
Thank you. Yeah. Whoop.
For those folks online, thank you so much for being here. We're going to end the presentation now.